Thank you to JCI Insight for allowing me to present this author's take on behalf of my co-authors. My name is Dr. Mark Geyer, and I'm a member of the Leukemia Service and Cellular Therapeutics Center at the Memorial Sloan Kettering Cancer Center in New York City. Today we'll be discussing our recent publication on safety and tolerability of conditioning chemotherapy, followed by CD19 targeted CAR T cells for relapsed and refractory CLL. Chronic lymphocytic leukemia, or CLL, has a considerably heterogeneous natural history, with some patients never requiring therapy or only needing treatment after a long interval. Oral molecularly targeted therapies have transformed the therapeutic landscape in CLL, but some patients remain at high risk for eventual progression on targeted therapies, including those with loss of TP53 and heavily pretreated patients. Those with progression on ibrutinib, idelalisib, or venetoclax have a particularly poor prognosis. Chimeric antigen receptor modified T cells, or CAR T cells, combine the specificity of an antibody with the effector functions of a T cell. The target antigen in this case is CD19. CD19 expression is restricted to B cells from early in development until differentiation into plasma cells. Several generations of CARs have been described, with second-generation CARs like the one studied here providing signal 2 on target engagement through the addition of cytoplasmic signaling domains of T cell costimulatory receptors. The CD19 targeted CAR construct we used in this study utilizes a CD28 costimulatory domain, and I will refer to the construct as 1928 zeta. To make CAR T cells, a patient's own T cells are collected, activated, transduced with the viral vector, expanded, and then cryopreserved until ready for infusion into a patient. MSKCC first began treating patients with purine analog refractory CLL using CAR T cells in 2007. Of note, many patients enrolled to the study were exploring multiple therapeutic options, and several ultimately pursued alternative therapy. 3 patients died prior to CAR T cell infusion. 27 patients ultimately received 1928 zeta CAR T cell therapy on this study. Please refer to Figure 1 for full details. We reported on 20 of these patients in the article, namely the patients with CLL or other B-cell non-Hodgkin lymphoma. I will not be discussing patients who received additional cellular therapeutics in addition to 1928 zeta CAR T cells, or the patients with Waldenström maculoglobulinemia. This study was conducted in stages. The first 3 patients with CLL were treated with 1928 zeta CAR T cells without conditioning chemotherapy. The subsequent 4 patients received cyclophosphamide followed by CAR T cells. The study was then modified to allow investigators' choice of conditioning chemotherapy, enrollment of patients with other B-cell non-Hodgkin lymphomas, and therapy with ibrutinib concurrent with T-cell collection and CAR T-cell administration. The schematic at the bottom describes flow through the clinical study after enrollment. Patients reported in this study were heavily pretreated, with a median of 4 prior lines of therapy in the CLL patients and 8 prior lines of therapy in the patients with B-cell non-Hodgkin lymphoma. High-risk features including TP53 loss, complex karyotype, and unmutated IGHV were noted in subsets of patients with CLL as listed. Most of the CLL patients were purine analog refractory. CLL cells exploit a variety of mechanisms to escape elimination from the endogenous immune system, including induction of T-cell dysfunction. The proliferative capacity of T cells in patients with CLL lags behind T cells in patients with other hematologic cancers, including ALL and multiple myeloma. One series from the University of Pennsylvania examined changes in the proliferative capacity of T cells in patients with CLL treated with ibrutinib. In patients receiving at least 5 28-day cycles of ibrutinib, T cells showed near normal proliferative capacity and decreased expression of immunosuppressive markers. In 3 patients treated with ibrutinib prior to CAR T cells, better CAR T cell expansion was observed, even though ibrutinib was held prior to CAR T cell therapy. The same report found that in xenograft models of B-cell tumors, better CAR T cell expansion and survival were observed in the mice continuously treated with ibrutinib as well as CAR T cells, suggesting additive, if not synergistic activity. These preclinical studies comport with the hypothesis that ibrutinib might repair T cell dysfunction in CLL. We suspected that ibrutinib might have influenced autologous T cell expansion, CAR T cell phenotypes, and clinical outcomes in our experience to date at MSK. 5 of our patients in this report were on ibrutinib around the time of CAR T cell infusion, of whom 4 were also on ibrutinib at the time of leukapheresis. The ibrutinib naive patients formed a logical control group. T cells from the 4 patients on ibrutinib at leukapheresis demonstrated greater cumulative fold expansion at all time points compared to the patients not on ibrutinib, with similar manufacturing time. Note the log scale in the figure at the right. Patients on ibrutinib are marked in blue. Specifically, median fold expansion at day 11 was 143-fold in the patients on ibrutinib at leukapheresis, compared with 26-fold in the patients not on ibrutinib, and median final expansion was 374-fold in the patients who were on ibrutinib at collection compared with 113-fold in the patients not on ibrutinib. Additionally, in patients on ibrutinib at T-cell collection, a greater proportion of end-of-production CAR T-cells demonstrated a central memory phenotype. Co-expression of CD62 ligand and CD127 was seen in 29% of CD8 CAR T-cells in the patients on ibrutinib at T-cell collection, compared with 4.3% in the patients not on ibrutinib. Of note, preclinical studies have suggested that CD8 CAR T-cells with this central memory phenotype in particular have the greatest proliferative capacity and ability to enhance survival as a single CAR T-cell subset. Peak CAR T-cell expansion in the peripheral blood was greatest in the two patients who achieved MRD negative CR, both by flow cytometry and by qPCR. Note the log scale in the qPCR graph. In the two patients achieving MRD negative CR, peak expansion was observed at 7 to 14 days following CAR T-cell infusion. All patients in this study developed fevers following CAR T-cell infusion in the context of cytokine release syndrome, or CRS. Fevers tended to occur earlier in the patients with ibrutinib exposure around the time of CAR T-cell infusion. The protocol was designed such that patients could receive 1/3 of the planned CAR T-cell dose on the first day of infusion and 2/3 on the second day. Of 11 patients for whom split-dose infusion was planned, fever led to the second day of CAR T-cells being held in 4 of 5 patients who were ibrutinib exposed, compared with 1 of 6 ibrutinib-naive patients. 2 patients, both with ibrutinib exposure, developed severe cytokine release syndrome and required vasopressors. While 9 of 20 patients developed neurologic toxicity, only 2 patients developed grade 3 neurologic toxicity, in both cases consisting of reversible encephalopathy. These 2 patients received corticosteroids for the treatment of their neurologic toxicity. No patients in this study developed cerebral edema. I invite you to review the supplement to review comprehensive patient-level details of CRS and neurologic toxicity in the patients reported in our study. Among the 12 evaluable patients with CLL receiving conditioning chemotherapy, 3 achieved complete response by IWCLL criteria and 2 achieved MRD negativity. 3 additional patients had an overall best response of stable disease, but exhibited partial or complete response in the bone marrow. A trend toward greater probability of objective response was seen in the ibrutinib-exposed patients. 3 patients received a second infusion of CAR T-cells, including 1 of the ibrutinib-exposed patients who achieved partial response in the marrow following the second infusion. None of the 3 patients who achieved CR have relapsed and remain progression-free at a median of 53 months. Of note, the ibrutinib-exposed patient who achieved MRD-negative CR eventually came off all therapy at 13 months post-infusion and remains in durable CR. This study joined several others reporting on second-generation CD19-targeted CAR T-cell therapy for patients with relapsed or refractory CLL. Only a minority of patients have achieved durable CR in most series, including this one, with rates of CR ranging from 20 to 35%. This stands in contrast to the reported experience in B-cell ALL, in which much higher rates of CR have consistently been observed. We and others are working to determine which patients with relapsed or refractory CLL are most likely to benefit from CAR T-cell therapy, and to develop strategies to overcome the inhibitory CLL microenvironment and enhance CAR T-cell expansion and efficacy in CLL. In summary, conditioning chemotherapy followed by 1928 zeta CAR T-cell infusion was acceptably tolerated at CAR T-cell doses much higher than the maximum tolerated dose in patients with ALL. Fevers were uniform after CAR T-cell infusion, but severe toxicity was thankfully uncommon. Similar to other series, approximately 25% of patients achieved durable complete response, and 2 patients herein achieved MRD negativity. Ibrutinib may enhance T-cell expansion ex vivo and may modulate end-of-production CAR T-cell phenotypes. Further investigation of CAR T-cell approaches incorporating ibrutinib is warranted, and broadly, the field needs to develop and investigate novel strategies to overcome the present limitations of CAR T-cell therapy in CLL. Thank you again for joining us for this author's take, and don't hesitate to contact us with any questions or comments.